So again, this section on genetics is about the little bit more complicated things, the different ways you can inherit things that give you non-Mendelian ratios and so forth. Before we get to that, we'll talk about the two Mendels laws and our now modern understanding of them, and that's it. So Mendel's laws, we got segregation and the other one, and they have to do with two different things, believe it or not. They're always confused in my mind, I don't know about yours. One of them has to do with separating the traits that make up, the genes that make up a single trait, and one of them has to do with divvying up the genes that correspond to multiple traits. Okay? In both cases, the law is that they will act independently, but we'll look at the mechanism underlying why. So for the law of segregation, the genes that correspond to making up a trait will have an equal chance of being partitioned into each gamete. So we start with our diploid sex cell, our spermatocytes and our oocytes. As they undergo meiosis and they divvy up their homologous chromosomes, if there is a gene version A and a gene version B, and you're looking at their descendant mature gametes, there is no reason to expect that gene A would be preferentially put into one cell over gene B, gene version B. Okay? So we're talking about a single locus, a single gene, but for which we have two versions. They are on two different homologous chromosomes. Okay? So it's chromosome A, but it's chromosome A version 1, chromosome A version 2. There's a gene on each one of those. Each of those has an equal chance of being partitioned into a cell. Okay? I've said it 3 times, we can move on. So independent assortment, we need to think of the law of segregation as being monohybrid, and this is being dihybrid, because those are the models we have our hands on. But the law of independent assortment concerns how you divvy up every single trait within the genome. Okay? And the point is that because genes are distributed across different chromosomes, we can assume that if you have such and such traits, such as color, is that the one that's on here? Yeah, so we got yellow seeds and green seeds. The genes for color are on one chromosome, but the genes for round seeds and wrinkled seeds are on a completely different chromosome. Therefore, if you inherit one trait, say yellow seeds, there's no reason to expect that you would also inherit round seeds every time that you saw the yellow seeds. Okay? The two genes are on different chromosomes, and because of the first law of segregation, they're equally likely to go into different cells, and the chromosomes are not linked. So what happens when two genes are on the same chromosome then? This is the important quote-unquote exception. Furby, give us a thought. Say again? That's one way to call it, right? They should travel together. If we're only talking about chromosomes as a holdup. Now, as a rule, what you just said does not happen. Why? Everybody write down an answer. So the class answer is crossing over. Yay. Now, there is a set of genes that will travel together on a given chromosome, despite crossing over, mixing and matching all the different chromosomes. So when do genes travel necessarily together? Specifically different versions of a gene. They're on the same chromosome, and they might not have covered this part. No. Say again?
They're linked.
They're linked, yeah. What does that mean?
We need one expressed to have the other.
One expressed to have the other. No, we're talking about the actual. We can get very complicated with that in a very big hurry. That's not what I'm looking for. But linked is a great word. They're linked because they're actually so close on the chromosome that crossing over in between the two genes is very infrequent. Okay? So crossing over happens as a function of distance between genes. If you get to a certain point, genes essentially sort independently, even though they're on the same chromosome. Okay, so that's all the fine points. But here, when you're studying this, go back and remember, independent assortment is based on chromosomes containing different sets of DNA, therefore different sets of genes. And if a gene is on a different chromosome, there's no reason that it should travel with another. If it is on the same chromosome, the simple answer is it should travel together. The more sophisticated, nuanced answer is that crossing over often prevents even that linkage from being observed. Okay. When you get to genetics, they do this chromosome mapping stuff, which gets into the incidence with which genes recombine and travel with each other, and that's all very neat, but I think you'll have to wait for it. Okay, incomplete dominance is easy. This is the intuitive one that we did in our backyard, right? Red plus white expresses pink. Okay? So if you have 2 copies of red, you get red. If you have 2 copies of null, you get white. So what I'd like you to start thinking about this kind of phenomenon as is that a lot of genes are quantitative. So in terms of how much pigment you make, well, in terms of the color that you witness, the amount of pigment that you make matters. So if you have two genes operating at a normal level, they'll give you red. But if you only have one, you get the intermediate state. A lot of times there are compensatory mechanisms such that you only need one functioning version of a gene, and we typically call that a dominant gene. If you think of all your enzymes, you think, oh, my body in every cell needs to be able to convert substrate A into product B. One copy of an enzyme will typically do, unless it's something that you have to make lots of. So you can take this back to your enzymes as well. But for now, you want to look at things that give you an intermediate phenotype given a hybrid genotype. That's incomplete dominance, not to be confused with codominant. So, like I said prior, human blood types A, B, and O are going to give people fits. So it's a phenomenon of codominance, and it's a phenomenon of multiple alleles. There are 3 different alleles in this blood system. This is not the only blood system that matters. Okay? So what these are a string of sugar residues attached to the surface of your blood cells. And what this does is provide a marker for your immune system to recognize and or attack these cells. So if you are a type A blood type, you bear a certain set of sugar residues on the surface of each one of your red blood cells. And your immune system knows it. So it says, hey, this is okay. It's cool. It's A. He's with us. But if you introduce something with the B series of sugar residues attached to it, that antigen, quote unquote, your body will recognize that as a foreign substance and attack and destroy it. In the process, it will engage a huge immune response, and not only are you anemic, but you're swelling up and causing all sorts of other problems. Hence, blood transfusions were thought to be impossible until very recently, until we discovered blood types like this. So what is O? Do we know this? Everybody write an answer down real quick. Okay, in a murmur, what is O? 1, 2, 3. Universal donor, I heard, which is true. It doesn't have any antigens on it, so it has neither A nor B. So this explains it. If it's not presenting these antigens, neither an A-type person nor a B-type person will attack an O-type. Okay? So what happens when you have this genotype, A plus O? You are what type? A. If you have B plus O, what do you have? If you have O plus O, what do you have? Is there any way to be O if you have another antigen? No. It will be dominated by the presence of that other antigen. Okay? So A and B together, you have both of those alleles, This is what we're calling codominant. The phenotype is you are immunogen reactive to that antigen. I know those are funny words, and it took me till grad school to figure out what they were. But if you have A, you're going to react to A. If you have B, you're going to react to B. So having both, you're going to react to both. Therefore, the phenotype is codominant. Note the nomenclature on here. This is what Sam was trying to suggest on the colors the last time. Because there are multiple alleles, it's best just to name the trait and use the different versions as a superscript. Okay? And I guarantee you there will be blood type questions on the test and in our exercises today. There will also be the MN and rhesus factor. Rhesus is a simple dominant. If it's there, it's there. If not, you need a double minus to not be there. Okay. So this is an interesting slide that I will not dwell on, but you're probably familiar with the XY system for sex determination in humans. 2 X's makes a lady, XY makes a guy, and one of the points is not everybody does it the same way. Moreover, other animals tend to have hermaphroditic states to go with their females. A lot of birds, for example, invert their methodology. They make the weird one, the weird hemizygous one into the females. And insects tend to just decide, hey, do I have enough DNA? Am I haploid? Am I diploid? Or do I have such and such ratio of sex chromosomes to autosomal chromosomes? And that's what will determine my gender. So I don't recommend trying to understand this at the outset, but know that we do it differently. Where it becomes important for us is considering that X and Y are separate from the autosomal chromosomes. Only X and X can cross over. So there's no crossing over for X and Y. And that's, moreover, if you have an X and a Y, you're restricted to your 1 X copy to cover all the genetic material necessary that's on X. We'll hit that again in the next slide. So here it is. So the classic example is hemophilia. And this is one of the first things we figured out was linked to the X chromosome. So like I said, if you only have 1 X chromosome and you got a bum copy of some gene on your X chromosome that you need to live, you're going to show the pathological state. You're going to show that phenotype much more frequently if you're a male than if you're a female. If you're a female, you have to get 2 bum copies together. So here's a quick exercise. Cystic fibrosis has a live birth incidence of 1 in 2,500. So 1 in 2,500 kids, at least of European descent, have cystic fibrosis. So, what is the frequency of that gene allele that causes that disease state in the whole population? I remember being asked this question. I remember getting it wrong. Write it out. What is it? I don't know what this slide is going to be like as a podcast, but let's just plug in our numbers, okay? Let's say, instead of dealing with CF, that we're talking about hemophilia. And the incidence of X chromosomes that bore this mutation was 1 to 25. How many would we expect to see? So, if you have a dad who's a carrier, that's what this denotes. The X chromosome has the H, which is the mutant. I'm sorry. So this system is set up correctly. So the male has to be viable. Okay. So X plus superscript capital H means he's got a healthy X allele. Notice that the Y does not carry this gene. The Y chromosome is a relatively useless piece of DNA. It's got something like 19 genes on it, 4 of which shut down the female program to make you a guy. Okay? So there's not a lot of content there. So this gene for hemophilia is on there. And if healthy dad meets healthy allele from mom, the children are viable. So the sons inherit the X chromosome from mom, which is also viable. The Y doesn't matter. And they were passed along 2 good copies. If dad passes along Y, but mom gives a bum copy, you get the disease because there is no corresponding hemophilia gene to cover for it on this X chromosome. However, if mom passes along bum gene and dad passes along good gene, we have a carrier state where they don't suffer from the disease because they have one gene that's good enough to cover for the other. So if we look at this and we say it's 1 out of 25, that means we can just simplify it. 1 out of 25 is a carrier. That means that mom has this genotype. Dad will inherently have this genotype because he has to be healthy. And so 1 out of 25 times the male will suffer. Whereas if you have to get, whereas women would never suffer because dad could never pass along the weak X chromosome. Okay. So it would go from 1 to 2,500 to 1 to 25 if we're on the X chromosome. That's the dramatic effect of a sex-linked gene. Moreover, men always suffer disproportionately from this because we just do not have the extra complement of X chromosomes to cover those genes. Color blindness is another great example of this. It's at the opsin for green or blue is on the tip of the chromosome, and it tends to get recombined through crossing over in naughty ways where one chromosome ends up with 2 or 3 copies of each opsin, and the other chromosome gets none. When you pass along the none, you end up not being able to detect blues or greens. Therefore, you cannot distinguish those wavelength colors. So, because that's on the X chromosome, we again disproportionately inherit that. Colorblind females are very rare. Okay, and finally, more than one gene can influence a trait. Skin color is a great example of this. This, as diagrammed, is essentially a tri-hybrid cross. And you'll notice that you need to get as many copies of these 3 contributing factors to get you to the darker phenotypes. So once we have completely heterozygous parents and we get together, it's only the rarest case that you'll have the darkest skinned children and only the rarest case that you'll have the palest children. Most of the phenotypes will add up. Each of their little bits of contributing concentrations of pigments will boost the color. And you could add that up as kind of a histogram as shown here. If that's not self-explanatory, then ask me later. But there is one cool thing about this that I read. If evolution required it of human beings, the conjecture is that we would re-evolve to either the palest white guy or the darkest black guy within how many years? What's your guess? Human evolution, what's the scale? Say, so there can be motivating factors for this. So white people eventually became white because we were in northern latitudes. We didn't have very much sunlight. We need the sunlight to make vitamin D so that we don't suffer from rickets. So we reduce the melanin in our skin so we can absorb more light. Dark people in more tropical areas obviously need to protect themselves from more sun damage, so they need to have higher amounts of melanin. So how do we, if we change environments, how long does it take to get from one to the other? Guesses? Shoot. A decade? So 1 generation. How many thousand years? 250,000. So the entire course of human evolution. Okay? Actually, twice the human course of Homo sapien evolution. Other guesses?
4 generations.
4 generations. 100 years. No. 10 years. We're getting to the scale. It's the guess is about 1,000 years. And the reason, if you think about it, that's pretty quick for an actual macroevolution phenomenon in a slow breeding population like humans. Every 12 to 20 years is our generation, right, in the native state. And the reason, this is something that you won't get to until grad school, but is really cool, so I really want you to listen to it now, is it's not just about what genes you code for, it's about how you control those genes. So most of these little inputs onto this chart here, they probably don't represent another copy of melanin. They probably represent something that binds the DNA surrounding melanin and says, go. And so if you have 3 things saying go, by golly, you make more melanin. And the reason it could change so quickly is because those are not actually nucleotide changes. We don't actually have to wait for the DNA to actually change those go or stop elements. What it is, it's a modification on how the DNA is wound that can be regulated by external stimuli. And the great example of this, they think that a large part of our diabetes now is actually because of these these changes to the structure of the DNA, not the actual sequence of the DNA. Epigenetic changes from in utero glucose exposure from mom eating junk food. So the external stimulus is mom eating junk food and therefore having spikes of glucose. The baby actually reads that information and folds it into how they're going to regulate their given metabolic genes without actually changing the DNA sequence. So the kid is destined to be fat at the start. Not fat, but to suffer that disadvantage in their metabolism. So there's a world of controls out there on your genes.